Take it away. Thank you all for being here and making time to be here. So, as Peter mentioned, I'll be walking you through my project, looking at uncovering proteins that mediate red activity in cancers using a tool that we call synthetic dosage lethality. So, just a little snapshot of what I'll be talking about. I'll give you a little background on what cancer is and the role that receptor tyrosine kinases play in cancer, and the focus of the Mulligan lab, which is on one specific receptor tyrosine kinase called RET. I'll walk you through our efforts trying to understand the molecular mechanisms that regulate RET activity in cancers, and my work using synthetic dosage lethality to uncover some of these mechanisms. So cancer, as you may be aware of, is characterized by uncontrolled proliferation of cells, and this is commonly driven by altered genetic alterations that alter signaling and metabolism within the cell. Now, one prominent class of proteins that's often implicated in cancers are receptor tyrosine kinases, and these are cell surface membrane proteins that act as signal transducers, meaning they convert extracellular signals into intracellular effector signals. And so they're most well known for their role in pro-growth signaling, as well as mediating other processes within the cell, motility, migration, and mediating metabolism. And so for these roles in the cell, they're often implicated in cancers through genetic alterations. Now, the focus of the Mulligan lab is with this specific receptor tyrosine kinase called RET. So RET, or rearranged during transfection, is activated following binding of a GDNF extracellular ligand to GFR alpha 1 co-receptors, which then recruit two RET monomers. Once these monomers dimerize, they undergo trans-autophosphorylation, which then is able to activate several downstream signaling pathways to mediate several effects, including proliferation, motility, and differentiation. Now, RET has a crucial role early in development. It's essential for development of neural crest-derived tissue, including kidneys, enteric nervous system, and some of the other tissue that's shown up there. However, activating mutations lead to ligand-independent constitutive activity of RET, which is implicated in several cancers. And so these alterations cause aberrant RET activity, leading to uncontrolled proliferation, as well as other oncogenic processes, including invasion and migration. And so these are some of the cancers that RET has been implicated in. So in attempting to or better understand RET activity in cancers, so we know the pathology of RET, and that's very clear, and is very well understood. However, the mechanisms that actually regulate or govern dysregulated RET activity in cancers is much less clear. So we have this very simplistic view of RET being active in cancers. It activates these downstream signaling pathways, and then there's a black box between that and this cancerous growth that we see. And so we do need a better understanding of exactly what's going on between RET activity and this transformation that we see in cancers. And to do that, we actually need to explore the sort of landscapes that are actually mediating or helping RET activity in these cancers. And we need something that's genome-wide, something that can survey all the proteins in the genome, all the genes in the genome, and help us identify exactly what's in that black box. And so the tool that I'm proposing to use is synthetic dosage lethality. So this is a closely linked concept to synthetic lethality, which many of you may be aware of. And this is a concept where if you have a normal cell, then if you inhibit a gene, we'll call it gene B, the cells remain viable. However, with synthetic lethality, where you have a loss of function alteration in your protein, it sensitizes cells to inhibition of gene B, such that when you inhibit gene B, those cells will die. And this is most well known for its clinical application, where PARP inhibitors are used to treat BRCA1 deficient tumors. However, not all alterations in cancers occur in tumor suppressor genes. They impact oncogenes as well. And so on the flip side, where we have synthetic dosage lethality, we have gain of function mutations. And this is where synthetic dosage lethality comes in, where if we have an activating mutation in one of your genes, then those cells are sensitized to inhibition of gene B. And so that leads to lethality. And this has been used to explore therapeutic targets for prominent oncogenes like KRAS, MYC, and PLK1. Now, In addition to its clinical utility, it's actually a very powerful tool that can be used as sort of a discovery platform for identifying or better exploring oncogene functions. And so we can actually use it to sort of fill in this black box or identify or shed light on some of the pieces within that black box by asking the question of if we have RET overexpression or RET hyperactivity in our cells, which genes are actually important or help RET mediate the survival or proliferation of those cells. And so in that sense, we can use it as a discovery tool to identify the players that are mediating RET activity. And another benefit of this is that the fact that it's genome-wide allows us to identify genes with a diverse array of functions, meaning they can regulate RET activity on both the transcriptional level as well as the functional protein level. And so we decided to form one of these SDL screens, synthetic dosage lethal screens. And the model cells that we used are the SHSY5 bineuroblastoma cells. And so we selected these cells as they have robust expression of RET. And we can actually model RET overexpression by treating the cells with retinoic acid. And so we can massively overexpress RET just by treating with retinoic acid. And treating these cells with GDNF, which is RET's ligand, we can actually turn on RET, and then we've shown that it actually is able to induce several RET-mediated cellular processes. And in that sense, it makes a very convenient model for modeling RET hyperactivity, which is what we do see in cancers. Now, in performing the screen, we had two conditions. We had our RET knockout cells and then our RET overexpressing cells. And the way the screen was performed was we used a genome-wide library of shRNAs. And these shRNAs will go and target, each shRNA targets a specific gene, target gene. And so when we infect these cells, we ensure that each cell gets only a single shRNA. And then we allow these cells to propagate for 16 days. And I'll walk you through an example here with this purple shRNA. For example, if that shRNA knockdown, that gene knockdown was non-lethal to the cells, those cells will stay alive and they'll be able to propagate over time. And at the end of the screen, we'll be able to detect that shRNA. However, if that gene knockdown was lethal to the cells, then those cells will die and you'll lose that shRNA signature from the pool of cells by the end. And so it's basically a dropout screen where you're looking for a dropout of specific shRNAs that led to lethal knockdowns in your condition. And so what we were looking specifically for are shRNA dropouts that occurred only in our RET overexpressing cells. And these would be indicative of genes that were essential for RET-mediated survival and proliferation of these cells. And now into the results of the genome-wide screen. So identifying hits from the screen, we started with over 20,000 candidates from the genome. The first step we did was identify the ones that were significantly underrepresented. So these were the ones that were significantly shown to only be lethal in our RET overexpressing cells. So the ones that had dropped out selectively in those cells. From those, we took the top 1,000 hits, so the most lethal hits, and we correlated their expression to that of RET. And by doing that, we can identify genes that are functionally linked to RET, as they'll have correlated expression. And from that 200, just above 200 list, I selected 25 candidate genes for initial validation of our approach. Now, as a proof of principle, I selected 2 candidates out of that initial 25 subset, and these are ASCL1 and NCAM1. And I'll explain this in a second, but I selected these 2 as they have predictable effects on RET activity. And so it allows for easier validation of the screen. And so here in the volcano plot, we can see where NCAM1 and ASCL1 stack up against the other candidates. We can see that they're among the top hits with the most lethal score, as well as significance. And just as extra validation, using another approach to identify candidates, and then comparing the top 200 candidates from each approach, we identified 70 genes that were overlapping between those 2 approaches, including ASCL1 and NCAM1. And so that tells us that these are very good candidates to pursue as proof of principle. So as I mentioned earlier, these have predictable effects on RET activity. And that's because ASCL1 is a neuronal transcription factor that's predicted to regulate RET transcription in literature. And NCAM1 is a cell-cell adhesion molecule, as you can guess. And it's interesting because it actually binds the same signaling complex as RET, the GDF-GFR alpha-1 complex. And so these two candidates represent two mechanisms of regulating RET activity, both at the transcriptional level as well as on the protein functional level. So now I'll give you a little rundown of my work investigating the relationship between RET and ASCL1. So ASCL1, or actinic homolog 1, is a transcription factor that's essential for establishing neuronal lineages. It's also essential for determining fate of neuroendocrine cells. However, it has implications in cancer as well, where it's responsible for expression of proliferation-related genes in neuroblastoma and lung cancers. In addition, it's also responsible for expression of differentiation genes in certain cancers to allow for redifferentiation of cancers, including some lung cancers and prostate cancers. Now, to actually investigate whether ASCL1 is able to actually regulate RET transcription in our model cells, I first attempted to generate ASCL1 shRNA knockdowns SY5Y cells. And so I used two different shRNAs that targeted ASCL1. However, I failed to generate a sufficient knockdown of ASCL1. And this was consistent with findings of other groups that we had reached out to. And so instead, I decided to generate CRISPR knockouts of ASCL1. And the way this worked was that I designed two guide RNAs that targeted exon 1 of ASCL1. And the plasmid that expresses those guide RNAs also expresses GFP, so we were able to sort single cells, single eGFP expressing cells. And from those, we were able to expand 96 clones, single cell clones, and we confirmed knockouts in 8 clones. And this is one of the clones. And so we can see that there's a really nice knockout of ASCL1 in our cells. In characterizing these cells, first I assessed their morphology, and there was no clear drastic difference in morphology between the ASCL1 knockouts and our wild types. And part of this work was done with help from Sanjay Adler, who's a MyTech student. So the first thing I asked was, is ASCL1 able to regulate RET expression in our cells? And using qPCR, I showed that there was an over 50% reduction in RET transcript levels following ASCL1 knockout. And we can actually nicely complement this with some immunoblot data. So we can see that when we do knock out ASCL1, we see about a 5-fold drop in RET protein levels. And so together, this told us that ASCL1 is able to actually modulate expression of RET. And what I'm currently working on is a rescue experiment by reintroducing ASCL1, see if we can recover RET protein levels, just to confirm that it's actually an effect of ASCL1. So the results of the SDL screen also told us that loss of ASCL1 is detrimental to RET-mediated growth. And so now we have two pieces of data telling us that ASCL1 is able to regulate RET transcription, and as a result, we actually lead to a result in abrogation of RET-mediated growth in these cells. And so my next steps would be to further characterize the functional outcomes of ASCL1 knockout on RET-mediated processes, and then try to rescue those by reintroducing ASCL1. And so I've identified ASCL1 as a regulator of RET transcription in our model system. This provides important validation that the SDL screen can actually identify transcriptional regulators of RET. And so it provides a nice framework for identifying other candidates from our screen that may be important for regulating RET transcription. So now that I've shown you sort of on a transcriptional level ways that we can regulate RET, we'll move on to the next candidate, which is NCAM1. So NCAM1, again, is a cell adhesion molecule. Its primary role is in cell-cell adhesion through homophilic interactions with other NCAM1 molecules and through heterophilic interactions with other cell adhesion molecules. It also has roles in cytoskeletal organization, so with actin organization around a membrane, in addition to serving as a signaling platform that stabilizes signaling complexes to induce different cellular processes through signaling pathways. So NCAM1 has a sort of broad expression pattern throughout the body. However, it's most known for its essential role in neuronal processes, such as regulating neuronal migration, proliferation, as well as synaptic branching and synapse formation. And for these reasons, it's implicated in several neurological disorders. However, it's also implicated in tumor promoting roles in several cancers. And so to assess how NCAM1 may be related to RET activity or regulating RET activity, I first generated NCAM1 shRNA knockdown SY5Y cells. And so we can see when I used shRNAs that targeted all major isoforms of NCAM1, I can see that there's a really nice knockdown of NCAM1. In characterizing these cells, we can see that they do have an altered morphology. And so they are a lot larger and flatter than the wild-type SY5Y cells. And in contrast to SY5Y cells that like to stick together and clump up on each other, these ones grow as a monolayer. And when we stain for phalloidin, we can see that in the wild-type cells that there's nice actin organization all throughout the cell. However, we lose that nice organization when we do knock down NCAM1. And we can see that there's more stress fibers forming throughout the cell. Further characterizing the cells, we can see that by using a proliferation assay, that these cells do grow slightly less or proliferate less than the wild-type cells. And if we take a closer look at their morphology, we can see that they have these bubbles in the membrane called membrane blebs, which are typically characteristic of apoptotic cells. And so to assess whether they were apoptotic, I performed a PARP cleavage assay. So PARP is cleaved in cells that are actively undergoing apoptosis. And what I saw was that, like the wild-type cells, there's no cleaved PARP in our NCAM1 knockdown cells. And we can see as a positive control wild-type cells treated with star sporine. So this told us that the NCAM1 cells do proliferate less, however, they are not apoptotic. So now to assess the relationship between NCAM1 and RET. The first question I asked was, does NCAM1 play a role in RET-mediated proliferation and survival, just like the results of our SDL screen told us? So to assess this, I used a live dead screen. And the way this works is that the nuclei of all the cells are stained in blue, and cells that are dead are stained in green. And so we can see that when we overexpress RET on its own in these wild-type cells, there's very minimal cell death. And we can see the same when we just knock down NCAM1. However, when we combine RET overexpression with NCAM1 knockdown, we see a drastic increase in the amount of cell death. And when we quantify this, we can actually see that there's almost a twofold increase relative to wild-type cells when we overexpress RET and knock down NCAM1. And so what this told us is that when there's too much RET, but NCAM1 is present in the cells are fine, they're viable. However, when there's too much RET and NCAM1 is absent, then that's bad for the cells and that leads to lethality. And so we asked the question of why is loss of NCAM1 lethal only in our RET overexpressing cells? So to investigate this, I first looked at hints in protein levels. And so I assessed first whether NCAM1 is able to modulate RET expression levels. And so we can see that following NCAM1 knockdown, we do see a reduction in total RET protein levels within the cell. And even when we overexpress RET using retinoic acid, we can see that there's a massive increase in our wild-type cells. However, we still have a lot less in our NCAM1 knockdown cells. And to assess whether this was a transcriptional effect, I saw no difference in RET transcript levels in the NCAM1 knockdown cells untreated with retinoic acid. However, when we treated with retinoic acid, we did see a small difference or slight difference. However, that difference is not enough to explain the discrepancy in RET protein levels between our wild-type cells and knockdown cells. So we have a large protein effect and a small transcript effect. So I asked the question, are other membrane proteins affected by NCAM1 knockdown? Is this a sort of global effect on membrane proteins in general? And what I saw was that with some cell surface receptors like EGFR, transferrin receptor, and ITG beta-3, integrin beta-3, there were no differences in NCAM1. So we didn't see a reduction in their total protein levels following NCAM1 knockdown. Nor did we see that in other cell adhesion molecules like N-cadherin and ion pumps, including sodium potassium ATPase. What's interesting is that we did find a difference in another receptor tyrosine kinase called efferent B2, and I will get back to this in a second. So now that I'm seeing that there's a reduction in total RET proteins following NCAM1 knockdown, I asked the question of, is RET still able to reach the membrane and be activated? So is it still able to mediate these processes? And so to address that, I used a cell surface vacillation assay. And the way this assay works is that you would treat your cells with biotin, and that would be conjugated to any cysteine residues on these surface exposed proteins. Now, biotin is membrane impermeable, meaning that only proteins exposed to the outside extracellular space will be tagged, and any proteins inside the cell remain unlabeled. And this allows us to actually purify any labeled proteins using streptavidin beads. And that allows us to then quantify and compare the amount of RET at the surface versus RET that's in the total RET in the cells. And so by doing that, I've shown that there, in addition to the reduced protein levels, that there is reduced RET at the surface of the membrane in the NCAM1 knockdown cells. And I did not see the same effect with integrin beta 3, nor with EGFR and transferrin receptor. However, I did see the same effect with efferent B2. And so that was the same protein that had reduced protein levels following NCAM1 knockdown, similar to RET. And so I asked the question of what could NCAM1 be doing to these proteins, and why these two? And so if we sort of look into them, they're both membrane proteins. They're both kinases that have roles in neuronal functions, whether that's neuronal proliferation, survival, differentiation, synaptogenesis, etc. And so that may suggest that NCAM1 has a broader effect on activity of membrane kinases with neuronal functions specifically. So now that I'm seeing less RET at the surface, I asked the question, is RET still able to be activated? And so I used a signaling assay, time course signaling assay, where I treated cells with GDNF, which is RET's ligand, to turn RET on over a course of 30 minutes. And so we can see that with the wild type cells that we can activate RET very rapidly, and there's high level of RET expression. However, when we knock down NCAM1, we see a drastic reduction in the amount of RET that's being activated, even when normalized for total protein levels. And so that told us that there's less RET at the surface and there's less of it being activated by GDNF. And so could this actually be a mechanism that's leading to the synthetic lethality that we were seeing earlier? Is knockdown of NCAM1 actually leading to reduced RET activation, which then kills these RET overexpressing cells? And so to assess that, I did a growth assay or a dependency assay where I seeded some wild type cells. I then overexpressed and activated RET and allowed them to grow for 3 days. I then added a highly selective and potent RET inhibitor called selpercatinib, and then continued to culture them for 3 days. And then I assessed their viability. And using this assay, I was able to determine that when we allow these cells to grow with RET hyperactivity, and then we inhibit RET, it leads to a significant increase in the cell death. And we do not see the same effect when we allow the cells to keep growing for a total of 7 days with just GDNF or normal cultural media, as well as when we just inhibit RET without RET overexpression. And so those were some nice controls to show that it's only when you allow the cells to grow with RET hyperactivity, and then you take away RET, that these cells will die. And so conclusion from this is that cells overexpressing RET do grow dependent on RET activity. And so taken together, we can see that in the NCAM1 knockdown cells, we have reduced RET total protein levels. We are seeing less RET at the surface, and as a result, we're seeing reduced RET signaling in cells that actually depend on RET activity. And so if you're taking away RET from these cells, that's leading to their death. And so it could suggest that NCAM1 is able to induce lethality in these cells by reducing the amount of RET that's available. So what's happening to RET? Where is RET going following NCAM1 knockdown? How is NCAM1 knockdown affecting RET activity or RET protein levels? And so this is some ongoing work that I'm working on. But there are some potential mechanisms that we can investigate. Some of these could be that NCAM1 is affecting the translation as well as maturation of RET before it's delivered to the membrane, which could lead to reduced total protein levels of RET. It could also be a membrane delivery defect. So between maturation and delivery up to the surface, there could be a defect in actually getting RET up to the surface. Furthermore, it could actually be a mislocalization. So whether NCAM1 is able to stabilize RET or stabilize RET in different compartments or at the surface would lead to mislocalization of RET, which would lead to subsequent degradation. And lastly, NCAM1 has actually been shown to serve as a stabilizer of FGFR1 at the membrane, and so serves as a platform for FGFR1 signaling, and the loss of NCAM1 in those cells leads to a reduction in FGFR stability at the membrane. And so we can see that there's a degradation of FGFR1. And so it's possible that NCAM1 may be acting in a similar way on RET in these cells. And so these are some of the things I am currently studying, some preliminary data with them. However, the most immediate next steps that I will be focusing on is to reintroduce NCAM1 as a rescue experiment to see whether we can rescue RET levels and RET activity and prevent that lethality in these cells. And then further investigate mechanisms of reduced RET levels through the mechanisms that I just mentioned. And so overall significance of this is that I identified NCAM1 as a novel regulator of RET activity in cancers and potentially a broader class of proteins that are involved in neuronal processes. I've also validated the screen as a powerful tool to identify proteins that regulate RET activity on the protein level. So looking at some of the overall significance, the work I've done has put a small dent in that black box. However, it's served as a important validation that this SDL tool can actually be used to help identify some of those players within that black box that help RET transform these cells or mediate these oncogenic effects. And so by building this framework, we'll better understand molecular mechanisms of RET-mediated oncogenesis, and we'll potentially be able to derive actionable targets, therapeutic targets for RET-involved cancers. And so with that, I'd like to thank my supervisor, Dr. Lois Mulligan, for all her support, our program director, Dr. Peter Greer, and my committee members, Dr. Lynn Posovit and Dr. Shalom Yang, for their time and support along the way. I'd also like to thank our collaborators, Dr. Franco and Frederick Vusukumar out of the University of Saskatchewan, as well as Dr. Matt Kurpi at the University of Ottawa. And I'd like to importantly thank all past and present members of the Mulligan Lab. Special shout out to Brandy Hyman, Dr. Brandy Hyman, for all her help and support along the way. And this project was funded by CHR. Thank you.
So I'm sure there'll be lots of questions, and we'll keep an eye on. I'll get my laptop up here so we can see who on the Zoom has questions. But go ahead, Shalom, go ahead.
Yeah, I'm very excited to see this.
Thank you.
Especially that you can project.
Yeah.
I was just wondering whether or not this going to have binding to the
Correct. So that was a question I did ask as well. And
question being?
Oh, sorry. The question was that is there an interaction between RET and NCAM1, association between RET and NCAM1? And so I did perform some. Present that one. References, sorry. Yes, okay, here we go. So I performed some co-IPs to see whether I can pull down RET with NCAM1 and then the reverse, so NCAM1 with RET. And so I was not able to pull down RET, NCAM1 with RET. However, with pulling down NCAM1 and then probing for RET, there was a very, very weak signal. So it's very unlikely that they're actually directly interacting together. So it's just not very convincing evidence there.
Second question is related to recent work.
Right.
Very exciting, we find one rat overexpressed, and then you knock down, they become lethargic, right?
Yeah.
And it's another gene. What do you think? What's happening?
With the NCAM, like that
Yeah,
modality?
when you're not overexpressed, right? You're not cell death. But when you overexpress, this you knock down another gene.
Yeah, so I tried to address that with the RET dependency assay. So I was able to show that cells that grew with RET hyperactive that were cultured over a span of 7 days, overexpressing RET, grew dependent on RET activity. And so when you took away RET, when you inhibited RET with sulfatinib, you were able to kill those cells. So that showed really nicely that these cells are dependent on RET activity. And so a potential mechanism is that because NCAM1 is reducing total protein levels of RET, that we are seeing a reduction in viability because we're taking away its dependency, that dependency on RET. In a way, yeah. I didn't think that.
They don't have that one protein. Okay.
Yeah.
Yvonne, go ahead.
Ava.
On slide 11, we were talking about one of the validation steps, which was to look for co-expression of your proteins of interest with RET.
Right.
So part A is, could you just explain exactly what you meant?
Right. So, yeah.
It was just
Yeah. So this was based on a concept found in literature. I found several studies that use this method of sorting genes, and it's that proteins or genes that are part of the same functional interaction network or functional network are likely to be regulated, similarly regulated as part of some sort of regulatory network. And so if you abrogate expression or activity of one of them, then you'll see changes in all the other ones. Not all the other ones, but some of the other ones. And so they're tightly linked through either expression or activity somehow. And so we use that as a filter to sort of identify which hits are actually or may be correlated to RET activity or expression at all.
So what is your data set?
Sorry?
What is your data set? Which samples are you correlating?
Oh, sorry. Yes. So we had RET wild type cells, and then we had RET knockout cells. So when we took away RET or RET knockout cells, we did actually a genome-wide or RNA sequencing experiment, and so that was done by Dr. Matt Krupe out of just the 2 cells, and we compared which genes were downregulated when RET was knocked out, and so those would have been rationalized to be part of some sort of functional network together.
So is that at all a concern that you could be disregarding genes that do have an effect on RET function, but not RET expression?
That is possible, yes. So it's likely that they're not part of the same regulatory sort of network of genes. Yes, that's a possibility. Yeah, good thing to think about. Sure, I thought of it at one point, but yeah, very good point.
So I can ask a bit of a follow-up to that because I was thinking along these same lines. With retinol, you get a very nice induction of RET expression with retinoic acid. Does escape also go up with retinoic acid?
So ACL1, it actually goes down with retinoic acid. So you can show that it's not a very drastic reduction, but if you culture it over time for a long period of time, this is in literature. They've shown that ACL1 is slightly downregulated with retinoic acid treatment.
But you believe that it is positively regulating RET at a transcriptional level?
Yes.
And is it known that it has cis-acting regulatory elements, the RET promoter region does?
Yeah,
Okay,
yep. So there's several.
Yes.
So the canonical sequence is an EBOX, and we find several EBOXes upstream of the RET promoter. And there's ChIP-seq data that supports that. And so you can actually identify specific motifs that ASO1 is able to bind to.
Does ASO1 bind to retinoic acid receptor, or is it in a complex?
So that's one thing I'd like to address with one experiment, treating the ASO1 knockout cells with retinoic acid and see whether that is able to recover ret levels, which would tell us whether ASO1 is involved in that regulatory network of retinoic acid-induced expression of ret. And so that's one of the experiments I'd like to do that I'm working on. However, there's no good rationale to support ASO1 and retinoic acid receptor signaling as well.
Well, I remember some stories years ago that NCAM1 associates with integrins in the context of axon pathfinding processes. Have you tried to look for co-localization of NCAM1 and RET or other players?
Yeah.
FP2?
So I actually just got trained on IF last month. So that's something in the works. Looking for localization, co-localization of NCAM1. First of all, I want to see where NCAM1 is throughout the cell, whether I can localize it to different compartments, and whether I can then co-localize RET and other sort of proteins along with NCAM1. And that can give us a hint of what that mechanism of NCAM1 and RET activity.
I was actually surprised. It was the NCAM1 knockout, knockdown cells that you saw clusters of cells.
Yeah, yes.
But you don't see that with these cells normally, do you?
No, not the wild types.
That seems counterintuitive, doesn't it? If it's mediating cell-cell interactions. Well, probably not so much, at least homotypic cell-cell interactions. Okay. Other question, David?
Questions about the cell model. So these SY5 cells, so these are neuroblastoma cells, correct?
That's correct, yes.
Do they have wild type or mutant RET?
They have wild type RET.
But they overexpress?
So endogenous levels are relatively high. However, we can actually model RET overexpression by treating with retinoic acid. And so they already have a relatively high baseline expression of RET.
Right. So you expose these cells either to retinoic acid or the ligand, or the ligand,
Yes.
So you can do both. Do you know whether then the signaling downstream is going to be similar? Because most tumorigenesis is ligand-independent activation, correct?
That's true, yes.
So do you know whether ligand-independent activation, when you get autophosphorylation, is that the same as you get with overexpressed?
So that's a very good question. I have not done any experiments related to that. However, sorry, I'm just trying to recall. So yes, yeah.
Getting more molecules on the cell surface. And so, I mean, you basically want to optimize dimerization.
That's true, yes.
So there could be things happening in the membrane as well, which will also facilitate that, not just the overexpression of RET.
Right.
Or it could be changes to the RET sequence, which is what happens with ligand-independent activation. And I don't know, maybe they all signal the same way, but maybe they don't.
Yeah, well, we do find different, based on things like localization and the types of mutations that you do get, or alterations, that there's small differences in the downstream effectors that may be activated. And so I don't have any information related to the system where you have wild type and overexpression. There are no alterations in that, in the domain where all the effectors will bind. So I have no information about differential.
I mean, thyroid tumors and small cell lung cancer.
Yeah.
I mean, they're exactly the same. So you kind of wonder whether are there different pathways that are driving oncogenesis in two very different tissues. Although they're both neuroendocrine tumors, I guess there's similarities.
Good point, yes.
So you used a RET inhibitor and showed that overexpressing cells are very, very sensitive to it. What happens when you start knocking down NCAM1 or knocking down does that change their sensitivity to retinoic acid or to RET inhibitor?
So I did treat these I did treat the ANCAM1 knockdowns with selpercatinib in that experiment as well. And I don't have the data with me, but there was also an increase in cell death compared to the wild types.
Yeah, so you'd think you'd get the same effect of
Yeah.
sensitizing them to
Yeah.
RET inhibition. We know from Fred talking about, there's a neat technique that he does where he goes and looks at transcriptomic data from lots and lots of tumors.
Oh yeah.
You know what I'm talking about?
Yeah.
So have you talked to him about looking for correlations between RET and CAM1 or Ski? Have you
Yeah.
already done that?
So that's a good point. I did try to look at co-expression or expression between, or correlated expression between these factors and other tissue types. I did, yeah, I did not.
You want to be concerned that this effect may be system specific. So doing it in other systems or just profiling that.
Right. So that's something I'm actually also currently investigating. All the way back down here. So I've done another neuroblastoma cell line, so SKNBE2s, and I can show that there's a similar reduction in RET protein levels following NCAM1 knockdown. There are other cell lines that I've identified that both express NCAM1 and RET at high levels that are not neuroblastomas. So things like SHIP77 and NCI, which are lung cancer models. And so these are ongoing work trying to see whether this is reproducible in
that's even better if you can manipulate it.
But I tried to do correlation analysis between RET and CAM1 expression, RET and ACO1, but it's not as straightforward as I thought. So maybe reaching out to Fred in the future would be a great idea. I won't go in there.
Other questions? If not, join me in thanking Montdher for the wonderful.
Okay.